Today I'm going to talk about this subject of iron overload and metabolic syndrome. It's a little bit of a new area for our lab, but I think you'll find it's very interesting. So anyway, there's a lot of evidence that, of course, the metabolic syndrome is a big problem these days, and that's been the focus of my lab for many years. So we've been working mostly on the role of different peptide hormones, such as adiponectin, in the pathophysiology of metabolic syndrome. And more recently, our research led us into studying the role of iron in the metabolic syndrome. So today I'm going to really tell you about some background information on this topic, about the effects of iron on adiponectin and insulin resistance and metabolic syndrome, and then spend most time on our recent work on this protein called lipocalin-2, or NGAL, neutrophil gelatinase associated lipocalin, which we believe is a very novel and important mediator of iron homeostasis and inflammation in metabolic syndrome. So, by way of introduction, we are all aware of iron. It's in our daily lives, and it's used a lot in terms of nutritional supplements. And the bottom line is you need iron, but iron overload can also have many detrimental and toxic effects. For example, there's an association of iron overload with the metabolic syndrome, and this has been realized for some time, but actually I think been underestimated in terms of its significance. The literature has also shown a bidirectional relationship. So like I said, you need iron. If you don't have enough, you have clinical complications, but certainly with iron overload, there are also clinical complications, especially cardiomyopathy. And clinically, there is now a well-defined dysmetabolic iron overload syndrome, a combination of iron overload with various signs of metabolic homeostasis being dysfunctional. So with that introduction, we believe that iron overload is appreciated but still very much underestimated as a contributor to diabetes and heart failure in the metabolic syndrome. And there's a lot of literature that I can choose to show you as examples of this. For example, this study showing that excess body iron was associated with higher risk of type 2 diabetes. Again, measuring serum ferritin level was positively associated with insulin resistance and metabolic syndrome, in this case in postmenopausal women. And it's also extremely important that we appreciate the role of iron overload in cardiomyopathy, especially in Thailand, where thalassemia is very prominent. And in fact, cardiomyopathy and heart failure is the main reason for death in thalassemia patients. So I'm going to show you a little bit of basic research next, where we actually looked at skeletal muscle, which is the main tissue which deals with glucose, as you know. And we examined the effect of iron overload on skeletal muscle insulin sensitivity. First of all, we just took cultured skeletal muscle cells and treated them with two different forms of iron. In this case, I'm showing you data with FeCl3. This is really just to show that over time, when we treat the cells with iron, there's an increase in intracellular iron concentration. And these are two different levels of iron. We also use this compound DPD, which is one of the available iron chelators. So you can see that it very effectively can actually chelate a lot of the intracellular iron. And this is used here as a basic research tool, but it's of course very relevant clinically because most patients with thalassemia or other forms of iron overload are treated with these iron chelators. And the last panel just shows a fluorescent way of measuring intracellular iron. So actually this is using a probe which is fluorescent green, but is quenched by iron. So the more iron in the cell, the less fluorescence. You can see that here, but when we use the iron chelator, the fluorescence is maintained. So with that model, we then went on to look at glucose uptake into muscle cells, and that's done using a radioactive tritium-labeled glucose. And the normal insulin response that you can see in the white bars is actually attenuated in the presence of iron. And that's at both 1 hour and 24 hours. So that's glucose uptake. We also measured insulin signaling, Western blot, for insulin receptor substrate, tyrosine phosphorylation, and AKT, threonine, and serine phosphorylation. All cases, the insulin effect shown very clearly here is attenuated by iron. So this is showing that iron caused insulin resistance. And as I mentioned, the use of iron chelators is very important clinically. So let's look at our basic research model and see how does the iron chelator affect iron action in muscle. As expected, you can see here, again, just let's look at this graph for glucose uptake. The normal insulin response shown here is attenuated in the presence of iron, but when that's combined with the iron chelator, we can see that there is less inhibition of insulin signaling. Another aspect we wanted to consider is whether this was reversible. So we can prevent the effects of iron with the chelator, but can we actually reduce the effects of iron by removing and withdrawing iron from the cell culture medium? And as you can see here, this is again just looking at intracellular iron. There's a decrease after 24 hours of iron withdrawal. And here's the normal insulin response in terms of AKT phosphorylation. It's reduced by iron here, but after 24 hours of iron withdrawal, it's restored again. So the good news is it's actually reversible. And as I said, this is very important because in thalassemia, especially prevalent here in Thailand, this is very important to maintain cardiac insulin sensitivity. So, and then we extended our work into cardiomyocytes for this reason, because the data I've shown you so far was in skeletal muscle, but one of the main reasons we're collaborating with groups in Thailand now is to study thalassemia. So now I'm showing you the effect of iron overload in cardiomyocytes, in primary cardiomyocytes actually isolated from the rat. And again, hopefully you can see here, looking at the insulin signaling by Western blot, that the normal insulin response is decreased after 4 hours, and especially after 24 hours of iron treatment of cardiomyocytes. So I won't go through all the details with you, but the effect is very clear. We also wanted to study the mechanism, and one very obvious candidate was reactive oxygen species, or oxidative stress. So first of all, we used several different assays using fluorescent probes to detect ROS production and show that in cardiomyocytes, the ROS actually was induced by iron treatment. This is just by fluorescence, and this one by flow cytometry. Same outcome, same data. And then this is some preliminary data, which is not too impressive in terms of statistics, but it clearly shows that, again, in cardiomyocytes, that here's the normal insulin signaling response. It's reduced by iron. And then when we use this antioxidant chemical, MNTBAP, which prevents oxidative stress, we can restore, again, the insulin signaling and prevent this effect of iron, suggesting that increased ROS is, in fact, a mechanism. So that's some preliminary data, unpublished data on the effects of iron overload in cardiomyocytes. And like I said to you in the introduction, most of our work for the last 10 years has been looking at the effect of peptide hormones, or adipokines, on the heart. And this is an example of a recent review, or several years ago, a review we published. And so in terms of the effects of iron in the heart, I've shown you that it causes insulin resistance, but we are also interested in whether it causes resistance to this hormone called adiponectin. Because adiponectin is also very important in terms of cardiovascular disease. Adiponectin is cardioprotective. Therefore, in obesity, diabetes, and metabolic syndrome, we lose adiponectin and we lose that cardioprotective effect. Therefore, it's important to maintain that cardioprotective effect if possible. So, we decided to study the effect of iron on adiponectin sensitivity. And the reason for this is that there's been some recent speculation that the phenomenon of adiponectin resistance might occur in metabolic syndrome, and in particular in heart failure. So this new phenomenon of adiponectin resistance is actually very important to characterize. So first of all, we proved that when we treat cardiomyocytes with iron, again this is using the probe, which is fluorescent green but is quenched by iron. You can see that in 2 dimensions and 3-dimensional view that in cells treated with iron there is less fluorescence. We also transfected cardiomyocytes with a reporter construct, which is an IRE-CFP. So it's a promoter which is responding to iron and inducing the expression of cyan fluorescent protein. And you can see here in cells treated with iron, there is more cyan fluorescence, showing that the iron overloaded cardiomyocytes is actually biologically active or functional. So then what was the consequence of this in terms of adiponectin resistance? Hopefully you can see here that we are looking at immunofluorescence using an antibody for phospho-AMPK. And phospho-AMPK is the main signaling target of adiponectin-mediated signaling. This is control, and this is cells treated with a small molecule adiponectin receptor agonist called adipiron, and there's an increase in the phosphorylation of AMPK. That's quantitated here in the graph, and in the presence of iron, actually we no longer see this increase in phosphorylation of AMPK in response to adiponectin or adipiron. So this is suggesting that iron is also causing adiponectin resistance in cardiomyocytes. And in terms of mechanism, I just want to show you that we investigated the role of FOXO1, which is a transcription factor which regulates expression of the adiponectin receptor, and itself is regulated by phosphorylation and translocation from the nucleus. And in the presence of iron overload, we actually found this increase in the phosphorylation of FOXO1, very clear increase. And so we think that this is part of the mechanism whereby there's a decrease in the adiponectin receptor expression and adiponectin resistance. So I'm going to summarize that first part of the talk and say that iron overload induced both insulin resistance and adiponectin resistance in cardiomyocytes, and that we believe both may play an important role in development of cardiomyopathy, in metabolic syndrome, and also in thalassemia. Okay, so for the second part, I want to switch to the story about lipocalin-2. So lipocalin-2 is a protein with 2 names, and basic research is always referred to as lipocalin-2, but in a clinical setting it's usually referred to as NGAL, or neutrophil gelatinase associated lipocalin. And it's actually now a very important diagnostic test for acute kidney damage. But we are interested in it because of its role in both iron homeostasis and inflammation as a root of cardiovascular disease and heart failure. And this is a review we published a couple of years ago now, summarizing the role of lipocalin-2 in iron metabolism and how this relates to the development of cardiomyopathy. So I'll introduce the main highlights of that topic right now. So basically, lipocalin-2 or NGAL is mainly derived from neutrophils, and it's part of the innate immune response. So it's very important in the body's defense against bacterial infection. And so the main mechanism of action of lipocalin-2 is to compete with bacteria for iron. And as part of the innate immune response, the lipocalin-2 prevents bacteria from obtaining iron from us, from the host. But in recent years, we've also realized that in obesity and metabolic syndrome, there's also a strong increase in circulating lipocalin-2 levels. And clinical studies have shown strong correlations of this increased lipocalin-2 with inflammation, dyslipidemia, and various forms of cardiovascular disease, including heart failure. So another review I want to point you towards is this one from a few years ago, where we actually commented on the the functional and mechanistic integration of infection and the metabolic syndrome, and in particular, the role of lipocalin-2. So let me show you how that work has developed in the last few years. I mentioned that there's a lot of clinical evidence for the association between lipocalin-2 and heart failure. And here's several examples from the literature. First of all, showing that serum lipocalin-2 concentrations are increased in patients with coronary heart disease. Even urinary lipocalin-2 levels can also be shown to associate with patients with chronic heart failure. And then even if we stratify the degree of heart failure by New York Heart Association classes, there is still a significant increase in lipocalin-2 within each class. So I think the available clinical evidence is pretty strong, and I now want to show you some of the work that we've done looking at the direct effects of lipocalin-2 in cardiomyocytes. And several years ago, the first thing we found was that treating cardiomyocytes, isolated primary cardiomyocytes with lipocalin-2, caused an increase in cell death. And that was shown by various ways, by tunnel assay and various mechanisms, various assays to measure caspase-3 activation. And in all cases, lipocalin-2 increased caspase-3 activity and cardiomyocyte cell death. So at that time, it was a totally new topic for us. We didn't know much about lipocalin-2, and we still don't know too much about signaling, because the lipocalin-2 receptor does not have a conventional cell signaling pathway. So by reading the literature, what we found was that the role of iron may be very important, because the main mechanism of action of lipocalin-2 in the innate immune system is to bind and transport iron. And when we started to look further into literature, we found a lot of papers showing that both iron overload and iron deficiency was strongly associated with heart failure. So here I'm going to just remind you of this fact of how lipocalin-2 works as part of the innate immune system. So normally bacteria can release siderophores, which capture iron and return to the bacteria with this iron, which is necessary for growth and survival. But what lipocalin-2 does is actually competes with siderophore, or actually binds to the siderophore iron complex and prevents it from being retaken up by bacterial transporters. So we wanted to understand the mechanism of action of lipocalin-2 and the importance of iron in the mechanism of lipocalin-2 action. But keep in mind that this siderophore is also present in the complex. Okay, so just a quick text summary before we move on to the detailed mechanism. So I've suggested that lipocalin-2 is essential in innate immune system, but we're also proposing that excess or prolonged lipocalin-2 induction has spillover pro-inflammatory effects that impact upon metabolism. I've also shown that the circulating lipocalin-2 levels are increased in obesity and in heart failure, various forms. And in summary of literature that I haven't shown that we've published in recent years is that using lipocalin knockout mice, these were more susceptible to bacterial infection, as you might expect, because there is no lipocalin-2 component of the innate immune system. These lipocalin-2 knockout mice were also resistant to high-fat diet-induced insulin resistance, and they were also resistant to stress-induced cardiac dysfunction. For stress, we used both pressure overload and ischemia reperfusion. So all of these suggest that the presence of lipocalin-2 is detrimental and contributes to the clinical complications in metabolic syndrome. And one of the things I'm going to focus on now to examine the mechanism of lipocalin-2 action in all of these is mitochondrial dysfunction. Okay, so as I mentioned, lipocalin-2 can come either as the protein itself or in that complex with the siderophore and iron. And these are called apo and holoapokylin-2. And we make these recombinant proteins in our lab. So just remember, as I go through the next few slides, the apolipokylin-2 is made such that it has no iron or siderophore bound to it. The holoapokylin-2 form is a complex of the protein plus the siderophore iron. So all three of them are complex. And I'm going to show you the different biological effects of these two different proteins in the next few slides. So don't worry about all the detail on these slides, but this is basically using Orberos system to measure mitochondrial respiration in primary cardiomyocytes. So it's basically measuring oxygen utilization. And the bottom line is that holoapokylin-2, which is the complex of protein, siderophore, and iron, but not apolipokylin-2, inhibited mitochondrial oxidative respiration in cardiomyocytes. So clearly there's different biological effects suggesting that it's not the protein, it's either the iron or the siderophore which might be responsible for these effects. And we also tested oxidative stress. So again, this is using a fluorescent probe to look at ROS production in primary isolated cardiomyocytes. This is a single cell under control and conditions treated with holoapokylin-2. And you can see in the quantitative fluorescence that there's an increase in ROS production with the holo form of apokylin-2, but not the apo form. And we found that when we use three different antioxidants, especially this, the general inhibitor IAC, but also this mitochondrial targeted antioxidant called SK2-1, we found that we could prevent the effect of holoapokylin-2 on ROS production, suggesting that it was acting, the holoapokylin-2 was acting on mitochondria to increase ROS production and decrease respiration. So how is that actually happening? As I said, the complex, the structure of holoapokylin-2 is a complex of the protein with iron and siderophore. So then we, first of all, looked at the role of iron to see if iron itself was important and had any direct effects. And then I'm showing you here the mitochondrial respiration and also the ROS production. But the conclusion is that iron does not inhibit mitochondrial oxidative respiration in primary cardiomyocytes. And also we showed that the effect of holoapokylin-2 was not attenuated using an iron chelator, which I introduced earlier in the talk. So this was a little bit surprising to us, and it led us to studying the effect of siderophore, bacterial siderophore. So we used these two structures, these two siderophores, to look at the effect on mitochondrial respiration by Orberos, by seahorse, and also on ROS production. And the conclusion was that these siderophores directly inhibited mitochondrial oxidative respiration and induced ROS production in cardiomyocytes. So it's suggested that the siderophore was actually a very important biologically active part of the holoapokylin complex. And that's a rather novel. There's only a few papers in the literature showing direct effects on cells of bacterial siderophores. We also translated the work to the apokylin-2 knockout mice. This is a mass spec showing the presence of bacterial siderophores with 2,3-DHPA here and 2,5-DHPA. You can see in the wild-type mice here, both are present, but in the heart of apokylin-2 knockout mice, there is certainly less of this 2,5-DHPA. And in correlation to that, when we look at the mitochondrial respiration in the heart of wild-type or apokylin-2 knockout mice, this is getting a little bit complex, but it's with and without ischemia reperfusion surgery. The conclusion again is that apokylin-2 knockout mice hearts have less of this siderophore, this 2,5-DHPA here. And in fact, those apokylin-2 knockout mouse hearts are protected from ischemia reperfusion-induced mitochondrial dysfunction. So So we believe that these studies identify therefores as novel regulators of cardiomyocyte mitochondrial function. And I think, hopefully you agree, that this may have widespread implications for various other tissues and diseases. So I haven't shown you all the data here, but I'm pleased to say this was accepted last month in PNAS, which is very nice. And some other work from our lab in Lapocalin-2 has shown activation of this NLRP3 inflammasome via TLR4 signaling. I won't go into all the details here, but this is also published in the American Journal of Translational Research. And two other very simple cell-based studies that we published on Lapocalin-2 really start to highlight an important role for autophagy in mediating the biological effects of Lapocalin-2. So Lapocalin-2 in muscle and in cardiomyocytes inhibits autophagy. So this is my conclusion. So therefores are regulators of cardiomyocyte mitochondrial dysfunction, and we think there's a novel axis of communication between infection, the microbiome, bacteria, and metabolism. Iron overload, we have shown, can also induce cardiomyocyte cell death, and it can also induce insulin resistance. Lapocalin-2 may be critical as a kind of node of interplay between infection and metabolism, and it binds iron, and so therefore, and like I said, probably it's very important in integrating innate immunity, the microbiome, or infectious bacteria, and inflammation and metabolism. And then I want to thank everyone who's contributed to the data I showed you today in my lab and also